All right. Hey there, zoology students. So today we're going to talk about the cells and structural components that sponges have. We talked about a little bit about sponges the other day, and now we get to talk about what they're made up of. So the essential question is, so please get this down in your notes. You should be taking notes over this, of course, today. The essential question: what types of cells and structural components do sponges have? You will see that these are going to be numbered. There's probably 10 of them, so just kind of sit tight. We're going to get through all 10 of them today, show you lots of pictures. Again, pause at any time you need to write additional stuff down. All right, here we go. All right, so the first type of structure or cell that sponges have. First of all, sponges have a division of labor. So they don't really have tissues, but they do have cells kind of working together and they have very specific jobs. The first cell is called a pianococyte. Pianococyte. And I know this is a cell. Can anyone figure out why we know this pianococyte's a cell? Because it ends in that's right, site right there. So that means it is a cell. So here's the definition of that cell. It's a thin, flat cell that line the outer surface of the sponge. It may contract and change the shape of the sponge or regulate water entry. I'm going to show you a picture of this in just a moment, but if you need to pause the video and get the whole definition down, you can. Really, the big picture is just they're flat cells that line the outer surface. So anything in like pink today is the most important thing. Okay, let me show you a picture of what a pianococyte is. Here we go. You're going to see this picture a lot. This is a cross section of a sponge. So we're like looking inside. This is the inside of the sponge and on the outer edge. And as you look on the outer edge, see these like, I don't even know what color you call this, like a green puke color, I guess. I don't know. But this green color here, those are all the pianococytes. And can you see how they're on the outer edge of the cell? So they're really, like this says here, it kind of lines, it's like the epidermis. It's the outer layer of the sponge, excuse me. Okay? This picture is found on page 6 in your packet. If you want to refer to it, you can. If not, not a big deal. We're going to go over this in great detail the next time we have class together. Okay? All right, so moving on to the next one. Number 2, another division of labor that cells have are called porocytes, and we know that this is also a cell because it ends in site. And these are tubular cells that lines the openings. I don't know if you remember us saying the other day, sponges are porous. They have like little tiny holes all over them. What makes these holes per se are the porocytes. The porocytes, so they're tubular cells that lines the opening. I'm going to show you what that looks like. Here we go. Boom, back to this one. You really can't see them, but they would be, it says porocyte here, but really they're cells. So they would be lining this structure right here and lining this structure right here. I have a better picture of it later on in the presentation, but they're the ones that line the openings. These little openings right here are the pores that sponges would have, okay? So you would kind of say this white area is where we'd find porocytes. That's where you'd find the porocytes, all right? Moving on to the next one. Here we go. Another division of labor is the mesohyle. So here's my question. Is this a cell? And you would say, no, it's not, because it doesn't end in site. It is a jelly-like layer below the pianococyte. So it's kind of like the gel-like stuff that sponges have so that the cells can kind of have a place to hang out. I'm going to show you that picture again. Let me show you what the mesohyle looks like. Here we go. We're going to go to it. So the mesohyle, here we go, or sometimes it's called mesenchyme, is the dark yellow. And you can see lots of different cells are sitting in it. The pianococytes, can you see, kind of sit atop it here on the outer edge. Okay? So this is just like the gel-like stuff. All right, moving on. Here we go. Boom. A fourth division of labor then are called amoebocytes. And again, these are definitely cells because it ends in site. And these are amoeboid cells that move in the mesohyle. And here's what they are used for. They do a lot. They have a lot of divisions of labor, these particular cells. definitely get down. Amoebocytes help with reproduction. These are the cells that literally can turn into sperm, okay? They also secrete structures, kind of help out the cell, and they transport food and they help with storage. Again, remember, sponges really don't have organs. So, and I said the word organ there. So, the cells kind of take over and try to perform certain things that the sponge needs in order to survive without having organs. So this cell does a lot. Let me show you a picture of it. It is the white cells. Can you see? And they would be in the mesohyal, and they kind of move around. They don't have a specific shape. They move kind of by crawling. That's what amoeboid means. They kind of crawl. But yeah, these are those white cells, the amoebocytes. Another name is the archaeocytes. We're not going to really call it that. We're just going to call them amoebocytes. Okay? Moving on, we're halfway through it. Okay, and then this is another division of labor. Is called the choanocytes. Choanocytes, or another name for them, are collar cells. Again, completely a cell because it ends in site. And actually, I would probably star this cell, put a big old star on it. You're going to see I'm going to talk about it a couple of different times. Choanocytes, probably the most important cells that the sponges have, and they're very, like, you only find choanocytes in sponges. Like, no other animal on the planet has these types of cells. What they are they're flagellated cells, which means they're cells with a flagellum that are below the mesohyal. And what they do is they line the inner chamber of the sponge. Why do they do that? The most important job is they create a water current, and they filter and they grab microscopic food. This is how sponges eat. This is how they eat. This is like, not that sponges have a mouth, but when the water goes in them, they grab food this way with their choanocytes, So these are one of the most important cells that we've talked about so far. And I'll show you a nice little picture. And we're going to spend a little bit of time really going over these cells when I see you the next class period, okay? Here we go. You ready? Let me show you a picture. The choanocytes right here. It's this whole entire structure, okay? So this is the cell. Coming off of the cell is this funky collar. Sometimes that's why they're called collar cells. And then in it is the flagellum. But this whole thing is the actual cell itself, okay? The whole thing is. So if you can imagine, as water moves through the sponge, then the flagella here grabs the microscopic food particles, and that's how the sponges basically eat. Put them in the mesohyal, and then the amoebocytes kind of transport the food everywhere. So the only way they can get the food is with these choanocytes. Very, very, very important cells. Very important. Okay, so here's just a summary of all 5 of the division of labors of cells and structures that sponges have. Just make sure you have all this information in your notes. We are halfway through because I told you there are going to be there's 10 structures total, so we have 5 more to go. Again, there's everybody now. All of those words, for the most part, should make sense. There's still a couple words we still need to talk about, and that's what we're going to do right now. Excuse me. All right, here we go. That's just another picture of a sponge. Now, this is a really nice picture, and I believe this is in page 16 in your packet. We are going to go over this in great detail when I see you next. But this, you can see again, these are the cells. Here are the pianococytes. Here we go. Here are those porocytes. See how they line the opening? They're actual cells. Here's a really nice picture of a choanocyte. And then these are the amoebocytes, and then, of course, the mesohyal. So it's just a nice little picture of everything together, okay? Again, we'll go over this. All right, a couple other structures that you need to know. So sponges do have something of a skeleton, and the 2 things that make up the sponge's skeleton are spicules, which is, that's what you're seeing right here. This is a spicule. It looks like the letter Y. Or a thorn. And so a spicule are thorn-like projections that provide structural support and protection. Probably the most important thing about a spicule that you might want to get down is they're made up of calcium carbonate. Calcium carbonate makes structure is hard. We find it in rock, but we also, check this out, calcium carbonate is found in our bone, in your bones. And this is one aspect what makes our bones hard is this calcium carbonate. So sponges have it if they have spicules, and this is what would make a sponge, hard to the touch. Other species, have a fibrous protein called spongin. It's right down there at the bottom, number 7. And so let's get this down again. Spongin is made up of a fibrous protein called collagen. And collagen, believe it or not, is also found in your bones, and it makes your bones somewhat flexible. So even though our bones are hard, they have some flexibility to them. And what sponges have is this collagen fibrous protein, and they call it spongin, and this is what makes sponges spongy. Okay, so there's lots of different types of sponges like we talked about the other day. Some sponges have a combo of the spicules and the spongin. Okay, some sponges are high in spicules, which means they'd be really hard, and some sponges basically just have spongin, which means those would be soft. Those are the sponges we're kind of interested in because they're soft and we could bathe our bodies with them. Okay. Back to this picture, and the only thing you can see in this picture now, can everyone see the spicules? They're the purple structures here. They're found in the mesohyal. You can see them, like under the microscope. Spongin you can't see. Collagen is a protein, so you can't really see that, but if the sponge had it, it would be in that mesohyal. Okay? All right, we're almost there. So, some other structures that sponges have, there's 3 of them. Can you see the 3 on here? One is called an ostia, one is called the spongocoel, and the last one is called the osculum. So, the ostia, going back to number 8, these are, we call them the incurrent pores. So ostia are incurrent pores. And what it says is choanocytes use their flagella to create water currents through these external pores called ostia. So this is how the water flows into the cell. It's called an ostia into the sponge. It's called the ostia or the ostium. Once the water is in the sponge, that open space that the sponge has is called the spongocoel. So the open space is called the spongocoel. And this is just like where food would be hanging out, oxygen, metabolic waste. But this is basically, the spongocoel is the center of the sponge. I'll show you a picture. And then finally, the osculum or oscula, plural. Usually sponges just have one osculum, and this is how water flows out. It's called the excurrent pore, and excurrent means out. So like this is the water flowing out. So this is the hole at the top of the sponge. So let me show you a picture of all 3 of these. Back to our picture that we always see. Here we go. So now we have talked about every single one of these words. This right here, even though it's not really labeled, I'll show you another picture. This would be the ostia, the holes. Okay. Can everyone see the spongocoel is that nice open space, and the big hole at the top is the osculum. So literally water flows into the ostia, hangs out in the spongocoel. If there's anything good, the choanocytes are going to grab the food, and waste products flow out of the osculum. Let me show you another picture really fast. Not that one, but this one. So like we've been looking, this is the inside of the sponge. This is the outside of the sponge. And so on the outside, you can see the little pores, okay? Those pores, the ostium, would be lined with porocytes. And then this is the big hole at the top, the osculum. So we have ostium, or ostia plural. Water flows in, goes to the spongocoel, which is smack dab in the middle, and then out the osculum. That just kind of shows you how water flows. And again, the choanocytes are always pulling the water into the sponge. And then last one is just going back to number 5, choanocytes. What you just have to know, they're super important. Here's a really nice picture of it. So again, here's the cell with the organelles. Here's that little collar that has a lot of little finger-like projections to trap the food, and this is the flagellum. Choanocytes filter microscopic food and trap it. They then place the food vacuole into these lysosomes, and then they digest it with help of the amoebocytes. So we're going to talk a little bit more about this when I see you. All right, so your job is to summarize. It says summarize the different types of cells and structural components sponges have. There might be 10, which means there really are 10. Like you should have 10. I labeled them out for you. You should have 10 structures. Some of them are cells that are making up our sponges. We are going to talk and review these structures in great detail come the next time I see you. So make sure you have all this information. All right. Thank you so much, everybody. I can't wait to see ya. See you real soon. Bye.